Today we are going to discuss about anti-ulcer, peptic ulcer disease. Peptic ulcer disease arises from the imbalance between defensive factors, mucous bicarbonate and mucosal blood flow, and aggressive factors, acid, pepsin, nasates, and Helicobacter pylori. Hydrochloric acid is secreted by gastric parietal cell due to the stimulation of hydrogen potassium ion ATPase, that is a proton pump. Histamine through H2 receptor, acetylcholine through M1 and M3 receptor, and gastrin are important stimulators of proton pump. Acetylcholine and gastrin exert their action directly as well as through release of histamine. Antral G cell produce gastrin on stimulation by dietary peptides, and that mainly stimulate release of histamine from enterochromaffin-like ECL cells and weakly stimulate proton pump itself. Parietal cells secrete hydrogen ion in the lumen through H plus K plus ATPase. Vagus nerve help in increasing acid by three mechanisms: direct stimulation of proton pump, stimulation of enterochromaffin-like cells to release histamine, direct release of gastrin by action of G cells, and inhibition of somatostatin by action on D cells, later inhibit release of gastrin. The main strategies employed for the treatment of peptic ulcer disease and gastritis are to neutralize gastric acid by antacid, decrease secretion of acid in stomach, increase protective factor like mucus and bicarbonate, protect the ulcer by forming a layer over it, stimulate the healing of ulcer, and kill H. pylori associated with peptic ulcer disease. Drugs for peptic ulcer, this is the classification: gastric acid secretion inhibitor, H2 antihistamine, cimetidine, ranitidine, famotidine, roxatidine, lefutidine, proton pump inhibitors, omeprazole, esomeprazole, pantoprazole, lansoprazole, labiprazole, dextrabiprazole, ipaprazole, prostaglandins analog, misoprostol, this is your gastric secretion inhibitor. And now it's a gastric acid neutralizer, that is prostaglandin analog, that is misoprostol. The ulcer protective, that is sucralfate, colloidal bismuth substrate, systemic and non-systemic antacids में आ जाएगा, जिसमें systemic में sodium bicarbonate, sodium citrate, non-systemic में magnesium hydroxide, magnesium trisilicate, aluminum hydroxide, magalgrate, calcium carbonate. और एंटी एच पायलोरी ड्रग में अमोक्सिसिलिन, कैलिथ्रोमाइसिन, मेट्रोनिडाजोल, टेनिडाजोल, टेट्रासाइक्लिन, और एंटी कोलिनर्जिक्स में पायरेंजीपाइन, प्रोपेंथिलाइन और ऑक्सीफेनोनियम. Regulation of gastric acid secretion. Terminal enzyme H plus K plus ATPase proton pump, which secretes H plus ion in the apical caluli of parietal cells, can be activated by histamine. Acetylcholine and gastrin acting via their own receptor located on the basolateral membrane of the cell. Out of the three physiological cytotoxics, histamine acting through H2 receptor plays a dominant role, because the other two, gastrin and acetylcholine, act partly directly and to a greater extent indirectly by releasing histamine from parenchyma enterochromaffin-like cells called histaminocytes located in the oxyntic gland. Why S2 receptor activate H plus K plus ATPase by genetic cyclic AMP, muscarinic, and gastrin. Cholecystokinin receptors appear to function through phospholipase C IP3 DAC pathway that mobilizes intracellular calcium ion. Cyclic AMP-mediated proton pump activation involves calcium ion. Secretomotor response to gastrin and cholinergic agonist is expressed fully only in the presence of cyclic AMP generated by H2 activation. As such, histamine participate in the acid response to gastrin and acetylcholine at more than one level, and H2 antagonists suppress not only histamine. Gastrin secreted from the antrum in response to rise in antral pH, food constituents, especially peptides, and vaguely mediated reflex involving ganglion cells of the enteric nervous system. The postganglionic enteric nervous system neurons elicit gastric release from gastrin secreting G cells by liberating acetylcholine as well as gastrin releasing peptide. Dominant muscarinic receptor mediating vagal response is of the M1 subtype, and its location on the ganglion cells of the intramural plexus. Parietal cells and ECL cells muscarinic receptors are the M3 subtype. In the next class, we will going to study about mechanisms of action. This, what I have taught you, is the how the peptic ulcers can be in the gut can be induced in the gut. It is like how the peptic ulcer causes the gut causes peptic ulcer. This is the pathophysiology of the peptic ulcer. So this is regulation of gastric acid secretion in the peptic ulcer as well. Like it's a terminal enzyme H plus K plus ATPase from that will secrete H plus ion, and it can be activated by histamine. Then acetylcholine and gastrin acting through these on their receptor located, that receptors are located on the basolateral membrane of the cell. Out of these three physiological circuits, histamine act through H2 receptor and play a dominant role. Uh, because the other two, that is gastrin and acetylcholine, directly partly directly act, and to a greater extent it will indirectly by histamine from paracrine and enterochromatic cells called histanomonocyte that is are located in oxyntic gland. So H2 receptor that will activate the H plus K plus ATPase that will generate by that is H plus K plus ATPase is activated by generating cyclic AMP, muscarinic and gastrin. Cholecystokinin receptors appear to function through phospholipase C, that is IPC, that pathway that immobilizes the intracellular calcium ion. That will cyclic AMP is mediated by the proton pump activation that will activate the proton pump, and it involves the calcium ion. The secretomotor response to gastrin and cholinergic agonist is fully expressed only in the presence of cyclic AMP generated by H2 activation. As such, histamine participate in the acid response to gastrin and acetylcholine at more than one level, and H2 antagonists suppress not only histamine and gastrin secreted from the antrum in response to rise in antral pH, food constituents, especially peptides, and vaguely mediated refluxes involving ganglion cells, that is in the enteric nervous system. The postganglionic enteric nervous system neurons release gastrin release from gastrin secreting cells, and dominant muscarinic receptor mediating vagal responses of the M1 side. Its location is on the ganglion cells of the intramural plexus. Parietal cells and E cells are the M3 subtype. So in the next class, we will discuss about the mechanisms of action. Thank you.